racial profiling and discrimination.
People say, oh, racial profiling. Well, we profile people all the time. All the time. I walk through a place, they said he's Hispanic, I open my mouth, he has an accent, he's definitely Hispanic. That's profiling, you see? So it is very true that we're going to be profiling, but we are profiling you for a reason, for the purpose of knowing you better and trying to help you to have better health and eliminate these disparities. And what people do not understand is that at this point, it's a perfect point to do this type of studies. I think that people need to be educated. And I think that's the key for all of this, is education. You see, let's say, for example, that I have 5 pieces of DNA that are going to predispose me to have diabetes. I would rather know that I have those 5 pieces of DNA, and I'd rather find out how I can control my environment to avoid for me to get diabetes than not knowing. So the idea of profiling to me comes from fear, and fear comes from lack of education. Because at this point we have the tool to be able to identify the genetic information. What we are lacking, and this is where we need huge help, is the environmental components. That's where the limitation is. We need more people helping us, working together in developing measures of the environment and identifying what is environment in that specific trait or disease we're studying.
This question of will we benefit is a question that goes to whether it's in part a risk question, whether the risks of participating in genetic research will outweigh any potential benefit. And why do I ask these questions? First, because the history isn't good. And that is, historically, genetic information has, not very good genetic information, but it was believed to be good at the time. Genetic information has historically always been used in ways that adversely affected minorities, African Americans and other groups. It has always been used to show inferiority. It has been used to make some groups seem superior to others. That's just the way, the social utilization of the information. The second point is genetic information is very powerful information. And that is, we believe that genetics in this society, that genetics is predictive. And we know not a great deal at all about interaction between our genes and our environments, for example. We don't know very much about the interaction between genes and genes, as well as genes and environment. Nonetheless, we have thought of genetic information as very deterministic. So that if you have a negative association with a piece of genetic information, we have tended as a society to push that in the direction of saying something harmful or stigmatizing about a population. So with respect to our ethical issues, we are working against a tough history. And part of the effort has to be, can it be turned around? Can we use genetic information today in a way that's beneficial or positive rather than a way that's negative?
There's been a study in the relatively recent past of a particular combination of medications for treating heart failure. The investigators used basically self-identification of African American ancestry as the kind of indicator for individuals who might respond better to this form of treatment than others. I don't fundamentally believe that they think that that is the best indicator. I think they would probably, if you were to ask them, acknowledge that it's really just a kind of loose surrogate for other things. But what I guess happens is, because it's the easiest way to go and it's the one that's most likely to be easily translatable into some clinical activity, that's what gets used. And so whether it was intended or not, it's the message that gets communicated that somehow so-called race matters in terms of, in this case, making decisions about medication. I think most investigators, if you were to, if it's relevant to the research, would quickly acknowledge that it's a very poor indicator. We wish we had better, but that it tends to be what gets used because it's easiest.
There may be some markers that make certain drugs work better or worse for different people, and these markers, and many of these have been studied. Some examples are P450 phase 2 metabolizing enzymes. So there's a whole series of defenses that our body has against exogenous chemicals. And these have adapted to be different in different parts of the world because there's different plant species largely that we're exposed to in different areas of the world. So there are some of these differences that clearly suggest pharmacogenetic differences might exist in how people are responding to drugs. But if your groupings are really so large and heterogeneous, the likelihood of anybody in one particular group having all of the variants that may be even typical for that group, maybe the average person has those, is very small. In African Americans especially, there's a lot of admixture. I mean, there's some people that have more African ancestry and other people that have more European ancestry and others that have more Native American ancestry within this large group that you would call black or African American. So knowing or projecting a drug that would treat them, especially if it's a drug that is also then different in how other populations can utilize the drug or respond to it, is very tenuous. And I think if there are real differences that make the drug better for people with African ancestry generally, there's going to be some people in that sample that this drug is not better for, and they may be more European at those few loci.
I think what will probably come of this at some point in time is if there is a difference that's reproducible between self-identified African Americans and others as it relates to a particular medication, to me that means that there are probably a gene or maybe several genes that are represented at higher frequency in African Americans than others on average, and which are responsible for that difference. And the real challenge would be to find those genes, because in an ideal world, what you would do isn't to test for admixture, and it's not to test or just to use self-identification. It's to say that the reason, on average, African Americans respond better to this drug than somebody else is there's a greater probability that the following 1 or 2 or 3, whatever number it is, genetic variants are going to be found as greater in African Americans than somebody else. And what you'd really like to do is test for those specific things. Then you could do it in anybody. But I think we're still a ways away, and I view it as a stepping stone in a sense. The fact that self-identified race can show that there are differences in drug response is an important first step towards figuring out what the genes are that are responsible for that, and then eventually, from a clinical point of view, developing an approach to looking at those particular genes.
And really that's where everything is headed. So I think we're sort of at an intermediate point. Again, we have to think about snapshots in time. So here, this is kind of the second point in what I see as a progression from understanding that there are epidemiological differences to then quantifying those and seeing if they really are there and if they're biological or if they're cultural and how to understand those two things. And then moving on to finding the actual genes that determine those differences. And using those in a clinical situation is generally what we call personalized medicine. And it's kind of where people think we're headed with some of these diseases.
When a disease is found to be especially prevalent in a given group, the public has a tendency to oversimplify and conclude that the difference is genetic when it's more likely due to environmental factors such as differential health care. When genetic markers are found that identify race, the public has a tendency to conclude that the races are significantly different genetically, and sometimes employs this notion in a way that adversely affects minorities.